this topic because we did discuss some complicated stuff. So if you don't understand them clearly, it's completely fine. Just let me know while you are summarizing that these are some things I'm not very confident about, and we can go back and look at the fundamentals. So I talked about homologous, non-homologous chromosomes and a lot of other things, right? We started here. Yeah. Okay. Over to you. Let's go with a quick recap. And this recap is not like a test. So if you, while giving the recap, you think that something you forgot or could not remember or do not understand, just we can go over it quickly. It's a good practice that we revise the last class so that today's class makes sense to you. Yes, Aisha, over to you, whenever you're ready. Hello?
Yes, sir.
Yeah. You're mute, Aisha, if you're speaking.
So do I like give summary
Yeah,
of it?
just summarize it. Like, what did we talked about in the last class? What is a homologous chromosome? What is a non-homologous chromosome? What is a recombination, crossing over? So you see a lot of figures here. So just basically go over these. Yeah.
So we learned about linkage, where it is the physical association between two or more genes. And there are two chromosomes, one from the father, one from the mother. One will be dominant and other will be recessive. And
So it's not true about the chromosomes. So chromosomes are not dominant or recessive. On the chromosomes, there are versions of genes called alleles. So the chromosome that we got from maternal side might carry one allele for a gene. Similarly, the chromosome that we get from the father side might carry another allele for the same gene. Okay. Now, among those alleles, one can be dominant or one can be recessive. So it's not like on one chromosome, like I have represented here. It's not a very good way to represent, but this is a simplistic way to represent so that you understand. It is never the case that on one chromosome, all the alleles or all the genes are dominant, or on the other chromosome, all are recessive. Okay, so that the rule of dominancy and recessiveness only applies to alleles, but not to the chromosome itself. Okay, does that make sense?
yes, sir.
Yeah, that's good. So just Rabia has joined in. Hello, Rabia. Good evening.
Good evening, sir.
So Aisha is giving us a quick recap of the last class. If you feel that you want to pitch in somewhere and add something, please feel free to do that. Okay. Yes, Aisha, go ahead.
So we learned about the homologous chromosomes, which are similar in carrying the same gene on the same locus.
Okay.
And they have the same position on the centromere and same length of the arms.
Perfect.
And we learned about the sister chromatids and non-sister chromatids.
So what are sister chromatids and what are non-sister chromatids? Just like you told us about the homologous chromosomes. What do we understand by sister and non-sister term here? Are you there? Aisha, you're mute if you're speaking.
Yes, I'm sorry, my mic was on. So sister chromatids are the same DNA of a chromosome,
Yes, you're right. So sister chromatid, let me just polish your words only. So you mean to say that sister chromatid is just a duplicate of the chromosome, the other chromatid on the same chromosome, right? So when on the same chromosome there are two chromatids, it means that the chromosome has undergone DNA duplication. So sister chromatids will be exactly identical. They will carry the same alleles at the same location on the same chromosome, right? Two arms. Good. And what are non-sister chromatids? Aisha, you're again mute if you're speaking.
Non-sister chromatid are different copies of the. I'm not able to put it into words.
You see here two chromosomes. What are these two chromosomes that's in front of you in the figure? What are these two chromosomes called? Rabia, would you like to help Aisha here? What are these two chromosomes? Rabia, you are also mute if you are speaking. Am I audible, people? Hello.
Yes, I am.
Okay, great. So am I audible to you, Rabia? Because I don't know if Rabia can hear me and respond. Okay, yes. So Rabia can hear me, but you cannot speak. Your mic is not working. Okay. Was okay. Okay, so let me talk to you about this part. So the two chromosomes that you see here are called homologous chromosomes, right? So whenever you have to define something, just keep in mind the reference. So non-sister chromatids are two chromatids which belong to two different but homologous chromosomes are called non-sister chromatids. So one chromatid from this chromosome, anyone, and the other chromatid from this chromosome, anyone, because each chromosome has two chromatids. So if you are taking one chromatid each from each chromosome from a homologous pair, that is called a non-sister chromatids. And if you are taking both the chromatid from the same chromosome, that's called a sister chromatid. Does that make sense, Aisha and Rabia? Is it clear?
Yes.
Yeah. So would you like to write this down so that it's more clear to you?
Yes.
Yeah. So write it. The two chromatids of the same chromosome are known as sister chromatids. And for non-sister, you can say that a chromatid, two chromatids, each belonging to a chromosome, each belonging to one chromosome in a homologous pair, in a homologous chromosome pair, is called a non-sister chromatids. So the question that they can ask is, what is probable to happen? Okay. Can two sister, like it can come as a statement where you have to figure out the correct statement. And these kind of question comes on this topic where they ask you, it is possible that two non-sister chromatids can carry different alleles of the same gene. What do you think? Is this statement is correct or incorrect? Is it possible that two non-sister chromatids can carry different alleles of the same gene?
Yes.
Yes, it's possible, right? Is it
Yes.
possible that two sister chromatids can carry different alleles of the same genes?
No.
No, that's not possible. Does that make sense to you, Rabia? Okay. So, yes, Aisha. Then we talked about recombination and chromosomal crossing over. Would you like to quickly go over this? What do you understand from this? You're speaking, you're on mute, Aisha. Is my voice lagging because you people are taking too long to respond back? So one possibility is that my voice for you is lagging or is delayed.
Very voice is coming.
Sorry? Come again, Aisha?
Very audible.
It's audible.
Yeah.
Okay, good. So would you like to go over this colorful diagram that we made in the last class? What does this tell? Again, starts from the very basic. Start from what you see on the board.
Now,
I know that this, yeah. Yes, Aisha.
two chromosomes. Now,
Two chromosomes are
homologous.
Seems okay. These two
And
chromosomes are homologous. And what's happening here?
and one of the chromatids of each are crossing over,
Right.
and the alleles are getting exchanged.
So yes, so the alleles, so that part is called
Chiasmata.
chiasmata or crossover bridge, we call it. Okay. Rabia has joined in back. Rabia, can you now speak?
Sir, can you hear me?
Yes, we can. That's great. So nice to hear you. Yes. So would you like to help Aisha with this image? What are we doing? We see some chromosomes here dancing, doing some Bhangra kind of steps. So what is happening? This exercise of summarizing might be very frustrating in the beginning of the session, but as you go forward, it's very important because I'm pretty sure all of you are just like me. When I was in the class, I still does not used to study much, but when I was out of class, I did not used to study. So self-study is a myth in the beginning. So at least I'm trying to, in some manner, force a little bit of self-study on you, but only in the class, in the first 10 minutes of the class. So try to make sense of what's happening. I know that you know this. The moment I will start explaining, everything will start coming back to your memory because it's there somewhere. You just have to brush it up. So just bear with me. It's for your good. And even if you're wrong in summarizing anything, even if you say something totally incorrect, it's completely fine and normal. No judgments at all. So instead of staying silent and making this class utterly boring, biology is very boring when someone just teaches you like a story because it's actually a story of life, how life works, what happens. There's nothing interesting in it if you hear it as a story. There is a lot of interesting thing if you are curious to know. Okay. So I can just keep drawing chromosomes and keep telling you this goes where, this goes here. And it will make no sense. So we discussed something. What is happening? Why is it happening? Where is linkage in this whole picture? Why we call it recombination? Remember anyone? Rabia? Are you there, Rabia?
Yeah, so I'm here.
Yeah. So staying silent is the only sin you can commit in my class. The only punishable offense. Not to speak, not to speak up, but to speak incorrect or correct is equal. Come on, speak up, Rabia. There's no point I go today and tell you about pleiotropic genes or polygenic inheritance and sex determination, chromosomal disorders, where we don't understand this. It will be just another gibberish words coming out of my mouth and the class will be over. Makes no sense. Do both of you want that we skip this session and I should continue with polygenic inheritance? We can do that because these classes are completely democratic. If you want me to move ahead, we can just skip all of this. I don't want to make it difficult for you people. I can just go ahead. But I am on purpose making it difficult for you people so that difficulty in the beginning helps a lot when you see yourself excelling. Yes. Rabia, what do you want? Do you want to struggle with me over this, or do you want me to skip this and go to polygenic inheritance and you will read it on your own and figure it out? Because we have covered it in the last class. So what do you want to do? What is your vote? You can even write in the chat if you don't want to speak. Again, democratic. And Aisha, you also? Just write skip or let's continue to struggle. These two, you can just write these 2 points. And if you won't say anything, I won't say anything, we'll just figure it out. So you take all your time. I have all the time in the world. Yes, Aisha, Rabia, no one is speaking.
Sir, I'll go with the first option.
Okay. Okay, so
yeah.
okay, Aisha has expressed her vote. Okay, it's fine. Rabia, what do you want? Shall we skip all of this and go back to new topic? Or do you want to discuss this? You can write in the chat. Okay, so if Rabia wants to discuss, then Rabia, you have to give a quick summary. Struggle comes with struggle. Okay, I'll help you. Come on, it's good. So you see 2 chromosomes here. Do you think they are homologous chromosomes or non-homologous chromosomes? Rabia. Aisha, you also. Are these homologous chromosome pairs or non-homologous chromosome pairs?
Homologous.
Homologous. And how do you decide that? Why it can't be a non-homologous chromosome pair? Or what has to be different here to make them non-homologous pairs? Because the same genes are present at the same location in both the chromosomes, only the alleles of those genes are different. Okay, that's why we get to know that these are 2 homologous chromosomes. Okay, perfect. Now what's happening in the second step?
They both are crossing over.
Yes, 2 Two sister chromatids are crossing over or two non-sister chromatids are crossing over?
Non-sister chromatids.
No, non-sister chromatids are crossing over, right? And as a result of the crossing over, what will happen? And the allele pairs will be exchanged, right? And this whole thing happens during which kind of cell division process, mitosis or meiosis?
Meiosis.
Meiosis. So you people know everything, but you just need to be given options, right? So that's both good and bad. If you know something without options, when given options, it is easier. But do not rely that your knowledge only should be limited to be when given the options. So correct, crossover bridge happens, allele pairs get exchanged. Now another question for you people is, can sister chromatids cross over and exchange allele pairs? Technically, theoretically, or it is not possible for sister chromatids to cross over? What do you think logically? There's no right answer here. What do you think? You should not take this much time to. So there are two things. You people don't want to speak. It's again fine. I'll not force you. I can just go with a complete monologue for the next 1 hour. Again, you can write in the chat if you want a complete monologue, and we can cover a lot of topics because I can go with the pace. But if you people want to understand, then you have to discuss. That's one thing I do push students for.
Will you repeat your question?
Yes, my question is, can sister chromatids cross over and exchange alleles? Or do you think it's not possible for them and only non-sister chromatids can do that?
They can't, right?
They can't. Okay. Okay. And why do you think so? What prevents them from not crossing? Yeah.
Because they basically have the same genes, like the same
Perfect.
alleles, so they basically can't go.
Exactly. That's a very good point, but your reasoning is completely on point, that they have the same alleles. They are exactly duplicate copies of each other. But still that does not tell that why can't they cross over? Why can't one chromosome that looks like an X go and cross over among itself and start looking like this? Right? It can just cross over among it sister chromatids. But after crossing over, if these two cross over here, whatever will be exchanged will be the same allele, right? At the same location. So crossing over can technically, theoretically happen even between sister chromatids, but that crossing over won't result into any recombinants. Do you understand? So it's as good as no crossing over. Do you understand that?
Yes, sir.
Yes, sir.
But nothing prevents them that they cannot cross over. So the proteins, the whole. So how do these two non-sister chromatids cross over? Is because there is a whole bunch of machinery and proteins that allow them to come together, bind to each other. Then there is a protein that cuts the DNA strand and then joins it with the other strand. That's how. It's not like just a fictional crossing over or exchange of material. It is happening in reality. So you cut something from where it's exactly like how you cut and graft plants, right? You must have studied that in your class, like in your vegetative reproduction, right?
Yes,
So
sir.
all the proteins can work on sister chromatids as well. But as Rabia correctly pointed out, you understand the process now, that there will be no net result, no difference, because they carry exactly the same alleles. Okay. Only when crossing over happens between non-sister chromatids, it gives rise to recombinants, non-parental combinations, which are different from the parental combinations, as you can see here at the end as a part of exchange, right? So this makes sense, correct? Okay. We also in the last class figured out that the percentage. So in a normal Mendelian cross, that so any two genes that follow Mendelian genetics in a dihybrid cross in the F2, their phenotypic ratio has to be 9:3:3:1. And total 16 progenies if we take, and out of those 16, 6 have to be recombinants, right? Which if you convert into percentage, it comes somewhere around 37.5%. So around 37% recombinants you should get in a normal dihybrid cross if the two genes are following Mendelian genetics. If you see any deviation from that 37% recombinants, if you are getting less recombinants and more parental combination, then what we can say about those two genes?
They do not follow Mendelian genetics.
Yeah, so what do they follow? Why do they do not follow Mendelian genetics? It's obvious that they're not following Mendelian genetics, but why? What is the reason behind that? If there are more parental combinations in progenies and less recombinants in F2 generation, what could be the reason behind it? What is the reason that does not allow recombinants to form when two or more genes are inherited together?
Crossing over.
Linkage. It's simple, right? If two genes are linked together, then from whichever parent they come, they will always come together. So there'll be no recombinants or very less recombinant formation. The more the linkage, the less is recombinant. There was one golden rule I told you about. Linkage is inversely proportional to recombinants. Correct?
Yes, sir.
Yeah. So these are fundamental things. You are allowed to forget it, but then you have to again and again brush it up so that it gets into your long-term memory. It might take time and some patience, which I'm completely okay with. Okay, but what I'm not okay with is you forgetting but not brushing it over again. Okay, cool. So on the basis of which I explained you this diagram, which is in your NCERT, I have seen many schools and even in some board examinations have taken this exact same figure, changed a few things. For example, these parental and recombinant percentages, they change and they ask you which one follows Mendelian genetics and which one does not follow Mendelian genetics. So you can see here, cross B is following Mendelian genetics because you will not get to know what was the phenotypic ratio, but what you get to know is the percentage of parental and recombinant types. So you see that 62%, 62.8% parental type and 37.2% recombinant type is just like what we figured out for Mendelian genetics, right? 6 out of 16 are recombinants. So here also around 37% are recombinant. So which means this gene is normally following Mendelian genetics and there are enough number of crossing overs happening, which are, it's not like in cases where genes are linked, the chromosomes do not undergo crossing over. They do undergo crossing over, but other genes get separated and form recombinants, not the genes that are linked. Is that clear, Aisha and Rabia?
Yes, sir.
Yes.
So yeah. So, and in this cross A, you can see that the parental type is 98.7. So almost 99% of the progenies have what the parents had. And only 1.3% are recombinant, which tells us that the two genes that we started with in this cross are tightly linked to each other. Okay, make sense?
Yes,
sir.
sir.
Okay, perfect. Okay, so that was what we did in last class. Please, after every class, I give you some questions as homework. Try to solve that. If you do it daily, then when exams come, you won't have to spend sleepless nights. And believe me, it is true. Everyone tells the same thing to their successive student generation, but very few follow. I was also among the ones who did not used to follow, but if you follow, it's good. Sorry. Yes, let's go. So in today's class, I will be teaching you three things, basically three important things. One is we already understand now the concept of crossing over genes, alleles, etc. When Mendel did all his work, and people who came after Mendel like T.H. Morgan, Hugo de Vries, Carl Correns, von Schermack, they all were working on genes in monohybrid, dihybrid, trihybrid, tetrahybrid, all these. But in your syllabus, you just have to worry till two genes, okay? Two genic inheritance. But all the characters in animals or in plants are not just governed by one gene having two alleles. Okay, that's a very uh, it's common for most of the genes and characters, but there are certain characters that simply cannot be controlled by one gene having two different allele, one dominant and one recessive. So more than one genes are responsible for some characters. Can you guess which kind of characters these might be where one gene is not sufficient to control it? So I'm talking about polygenic inheritance. So in monogenic inheritance, yes, Aisha, you have an idea?
Skin color and eye color.
Skin color. Yeah, perfect. That's a very good example. And would you like to elaborate a little bit? Why do you think skin color is not controlled by one single gene?
Because there are a lot of different colors and gradients of skin color.
Because there are far more variations that than a single gene can give rise to, right? We already know that if a single gene, like in pea plant, there is one gene for height, it has two alleles, capital T and small t. In a heterozygous condition, it will be tall. In a homozygous condition, this will be tall and this will be short, right? So either the plant is tall or short. Even if there is incomplete dominance and it does not follow the law of dominance, in this case, it can give rise to medium in some cases. Not in pea plant, but let's say in some other plant, capital T is not completely dominant over small t, so there is incomplete dominance. So you can find a medium plant. But still just three phenotypes, right? You can't go beyond that. Even if there is codominance, again, 3 or 4 phenotypes, you can't go beyond that. But there are many characters where much more variation is seen. For example, skin color. Human skin color has how many phenotypes? Can you count those phenotypes? How many shades of skin color is present in humans? How many shades of skin color is present in a zebra?
Two.
Two.
Two. In a tiger?
Three.
2, 3. Okay. In a leopard?
2, 3.
Yeah. In an elephant?
1.
One.
Right. One. Like one is the darker blackish. The other is like could be less darker. But again, 1, 2. Let's say. Yeah. And but how many skin shades do you see in humans?
4 to 5.
4 to 5. So if I take all the humans of the world, can I just categorize them in 5 different shades of skin colors?
No, sir.
No, right? It's very much varied, right? The moment you will start thinking that this is the fairest person, you will find someone even more fairer than the fairest person you think, right? Same goes with the dark skin. So human skin color is not controlled by one single gene. Okay, it's controlled by more than one genes. Okay, for example, let's say there are 4 genes, which I name as, we figured out that there are around 4 to 5 different genes that contribute to human skin color. Let's name them A, B, C, and D. Okay, these are the 4 genes that control human skin color character. Okay, now how does skin color comes? What is the thing that gives color to the skin in humans? Yeah,
Melanin.
melanin. Okay, so skin color in humans, it's not true for all organisms, but in humans, it's because of melanin. Correct. Now, melanin is a, what is melanin? It's a pigment protein, right? It's a protein that forms a natural pigment and hence gives a color, dark brownish color. Similarly, chlorophyll is also a pigment protein. Xanthophylls are pigment proteins. Carotene in carrots is a pigment protein. So similarly, melanin is a pigment protein. Now, if it is a pigment protein, all these 4 genes must be involved in its synthesis. So let's say gene A also is involved in melanin synthesis. And dominant alleles for these genes, which is capital A, capital B, capital C, and capital D, they all give rise to increased melanin. That's why they are dominant. So when they are present, they express more melanin. And recessive characters, what they will do, they will not start forming white or a different color pigment. It is just the less amount of pigment that gives us a fair color. So recessive alleles, which is small a, small b, small c, and small d here, let's say all these decreased melanin formation. Okay, is that clear?
Yes, sir.
Now, taking it into combination, we have 2 alleles for every gene. So a person who happens to have this combination, A, capital B, capital C, capital Z, and capital D, which means from both the parents, it got all the sets of dominant genes for melanin formation. So what do you expect the skin color for such a combination? Dark or fair?
Dark.
Yeah, so this will be the darkest complexion that humans can achieve. And if someone has all the recessive genes, so which means very, very less melanin formation, so this will be the fairest skin. Make sense?
Yes, sir.
Now, everything in between will be one shade. So the moment you, from here, you find that someone has a heterozygous condition for one gene, but homozygous dominant for all other. So this will be slightly less darker, or you can say slightly more fairer until you reach finally here. You understand?
Yes, sir.
So all the combinations in between give rise to one particular skin thing. And most of the times, these polygenic inheritance genes are also affected in most cases by the environmental factors. Like skin color does not only depends on the genetics. So if you are born to a fair parents, but uh, if I send the person to a very hot nation near the equator in Africa or in the equatorial country like South India, do you think that the person will maintain its fair skin color?
No.
No. So what will happen? Why? Why is that? Because it has all the genes for a fair skin, but still the skin gets tanned. We call it tanning, or skin gets dark. Why does that happen? Any idea?
Because of the weather conditions, probably.
Yeah, just one thing, sun. So exposure to sunlight induces melanin formation in the skin cells. Okay, this pigment is very important pigment. So the countries that are situated near the equator, the Caribbeans, the Africans, equatorial countries like Sri Lanka, southern part of the India, all these countries, you will see that people are mostly dark skinned because this pigment is required in their skin cells to protect them from the UV component of the sunlight. Okay, it's like a shield, it's like a sunglasses for every skin. Okay, whereas countries which are up north or down south, they have fairer skinned people because sunlight is not very intense and direct, and for very long periods they do not see sun at all. So for example, the British Islands, the Europe, down Australia, etc. Does it make sense? Canada, US. So people are fair skinned there, people who have been there, who are natives to that place. But if a fair skinned person starts living in equator, then the person will see change in the skin color, which is induced by exposure to sunlight. Okay, so melanin in the skin prevents from vulnerability of skin cancer. Okay, if you do not have melanin in the skin and you are at equator, then there are more chances that your skin cells will get damaged by the ultraviolet rays, because UV is very energetic uh, electromagnetic radiation. You know about the Vibgyor spectrum, both of you?
Yes, sir.
Yes, sir.
So UV is ultraviolet. It is even shorter than the violet that we see. Vibgyor starts with violet, indigo, blue, right? The blue light spectrum. It's even smaller than that in wavelength. So if there is less wavelength, there will be, what about energy? For any electromagnetic radiation, if the wavelength is less, what will happen to the energy?
High.
More, yes. Red, on the other hand, has the highest wavelength, but the least energy. Okay, perfect. So you will therefore see longer waves like infrared radios being used for communication purposes, and very shorter waves like microwaves, like UVs, and all these for purposes of excitation, heating, cooking, etc. Okay, because they have more energy. So everything makes sense. So this is called polygenic inheritance. Another example is, apart from skin color in humans, is the height in humans, right? Height also follows the same rule. You will not find humans where you can place all of them either in 2 or 3 bins in the height. Humans range from 4 feet something inches to even 7 foot something inches in the world, correct? And height again is not 100% dependent on gene. Yes, there is a limit set by the gene. So your genetics determine to some extent how tall can a person or how short can a person be. But early childhood experiences, your activities, metabolism, nutrition, that also defines your height. Does that make sense, everyone?
Yes, sir.
Yes, so write down. Let's define polygenic inheritance. If a character is, or if the inheritance of a character, write down. If the inheritance of a character is determined by a combination of two or more genes, then it's called a polygenic inheritance. That's called the polygenic inheritance.
Introducing the newest member of the plant.
Okay, you can give examples like skin color in humans, height in humans. Anyone can think of an example which is non-human example? It's very interesting. Think about it. A non-human example of polygenic inheritance around you, just around you, because these are in textbooks. You will just remember it. But if you can think of something, then you understood this concept. Anything, people? You can say skin color, eye color, height, hair color, all of humans, but something which is not in humans, but let's say in plants around you. Or in animals. Okay, think about it. I'm giving you this as an homework to find some polygenic inheritances in plants and animals around you, your pets, the plants that you might have at your home, or you might have seen in fields, or you might have heard of in agriculture, etc. Okay, it's good to know that. Let's move forward to the next concept, that is pleiotropic inheritance. So this is polygenic. The other is pleiotropic inheritance. Now, anyone knows what is pleiotropy? P-L-E-O, or if there's an I as well, not sure. But anyone knows what is pleiotropy? Quickly.
A single gene is responsible for different characters.
A single gene is responsible for different characters, you say. Is that what you say? Sorry.
Yes.
Yeah. Yes, right. So, okay, just 1 second. Oh. Yes, it's shared again. Can you see the screen, everyone?
Yes, sir.
P-L-E-I should be there. Pleiotropic inheritance. Yes, you're right. So in this, it is just opposite to what we saw above. So there are multiple genes here. So polygenic inheritance is, in 2 words, multiple genes, but single character. Correct?
Yes, sir.
Excuse me. Pleiotropic inheritance is a single gene that controls multiple phenotypes, multiple characters. Okay. So this also happens. Okay. There are normal examples of this, but one very famous example that your NCERT also cites, let's first go for that, is a disease condition called phenylketonuria. Okay, it has 3 words. Pay attention to: phenyl, ketone, urea. So one thing that you can just figure out by this is it has something to do with the phenylalanine, okay, amino acid, and ketonuria means it causes a disease or disorder which also involves kidneys, and you can detect this through the urine. Okay. Now in phenylketonuria, what happens is, before we discuss phenylketonuria, which also we have to discuss as a disease, it is a genetic disorder, but we will spend some time here only so that both the parts are covered. How many of you know about phenylalanine? Phenylalanine. What is it? You have studied about this in, I think, class 11th, if I'm not wrong.
An enzyme.
Sorry?
It is an enzyme.
It is an embryo. Okay. Where it is in embryo? Phenylalanine. In what context? So it is not present in our body? Only in embryo? Yes, Aisha? You're not getting confused with placenta, right?
Yeah, I sent it in the chat.
Okay, that's great. You sent it in the chat. Enzyme. Phenylalanine is not an enzyme. See, names of enzymes mostly end with A's. That's one thing you should know in biology. It's a very handy trick. So most of the time you will find enzymes name, not all, there are exceptions for this, but enzymes mostly end with A's as a suffix. If you know any enzyme, just tell me. From digestive system class 11, do you know any enzyme? Amylase, salivary amylase, gastric amylase, right? Peptidase, hexokinase, glucokinase. Do you remember these enzymes? Lipase, nuclease.
Yes, sir.
Yeah. So Ase refers to that it's an enzyme. So all enzymes might not end with Ase, but everything that ends with Ase is an enzyme of one or the other kind. Enzymes are nothing but just biological catalysts, which are very similar to chemical catalysts. They make the reaction happen at a very lower temperature, pressure, and conditions, which normally will take more energy, more temperature, and more pressure. Just like when you add catalysts in a chemical reaction in your test tube. So phenylalanine is an amino acid. Now, do you remember what is an amino acid? So the motive is to prepare you for entrances. Okay. I cannot prepare you for entrances by just telling you some complicated stuff which you don't understand. That's as good as knowing nothing. So we will go through this difficult process of feeling that you might not know anything or you do not remember anything, but that process is very important. No one knows everything. Okay. You just have to keep discussing it so that it sets in your memory. What are amino acids? You have studied amino acids.
They're the blocks of protein.
Perfect. They're the building blocks of protein. You all have studied something called transcription, translation, and replication, right? The central dogma of biology.
Yes, sir.
Yes. So you know it. So in the central dogma of biology, DNA forms more DNA by the process of, how does DNA replicates?
Transcription.
No, replication. I already gave you the answer. DNA replicates through replication. Okay. Transcription is when you transcribe something into something else. Okay. So transcription is where DNA gets transcribed into a RNA. So whatever information it has, if you people have less confidence in your central dogma, please make this right now in your textbook. So DNA gives rise to RNA, and RNA gives rise to protein. Now, DNA can form more DNA by the process called DNA replication. Okay. RNA gives rise to, and DNA forms RNA by the process called transcription. And RNA forms protein by the process called what is this process called in the cell where proteins are formed? I think this you did in class 10th, where you studied the chapter cell. Then you studied cell organelles. One of the cell organelle was ribosome. And ribosome is known as protein synthesizing factory of the cell. Ribosomes bind to RNAs and form proteins through a process which is known as translation. Yes. Translation. Okay. So replication, transcription, and translation. For this translation to happen inside the cells, you must have made this figure also in your exams. So there is a bigger subunit of ribosome and there is a smaller subunit of ribosome. And in between the both goes your mRNA, right? And there are two sites in the ribosomes, A site and T site, where the tRNAs come and sit. And what does every tRNA bring with itself? It's called a tRNA because it's shaped like this in 2D. So this and it binds here. And what does it brings with itself to make a protein? It brings a amino acid, right? A long chain of amino acids is known as a polypeptide chain. And that polypeptide chain folds to make a protein, a 3D structure of a protein. So protein is nothing but a very long chain of amino acids. Does that make sense, both of you? These are fundamentals 101. If you people will not answer, I will not move forward. We will keep going back and keep revising it. Otherwise, this is a failed exercise. What is a protein, Rabia? Any protein, what is it? A protein is a long chain of amino acids bound to each other through peptide bond. Write it down. A protein is a long chain of amino acids. So amino acids are the building blocks of protein. So this one line we remember, we mug up, but we don't understand what it means. And if you really look forward to crack entrances, pursue your career as a doctor, or go into research, or make a career in biology or in sciences, mugging up can only take you so far. Just it might help you with some things, but to establish a career, you need understanding. Okay. So we have to work on the basics. So write down, a protein is a long chain of amino acids bound to each other through peptide bond. And now you will appreciate and understand if this bond between two amino acids is called a peptide bond. So what will the enzyme that breaks this bond should be called? Take a guess. Rabia? There's a bond called peptide bond. So when we eat protein, we want to break this bond so that we can absorb amino acids in our intestine, so that we can now make our own new proteins in the muscles. So what is the enzyme that is present in your intestine that break this bond that you have studied in class 11 in human digestion? Any guess, Aisha?
Something with pro. I don't remember exactly.
It's called peptidase. As simple. There is a peptide bond and there has to be an enzyme to break it. So it's called peptidase. Okay.
So what is proteolytic enzyme?
Peptidase. Peptidase is a proteolytic enzyme. The word proteolytic means it breaks down protein. Proteolytic. Lysis means breakdown in biology. That's where biology becomes easy if you understand words. That's why I always, in the beginning, I told you the way we are going to understand biology is to hit on the most difficult part of it, which is words. Words explain biology itself. Proteolytic enzyme. Proteo means protein. Lytic means breakdown. So there are some enzymes that break down protein. To break down protein, you have to break down these bonds, because a protein is nothing but a long chain of amino acids. The moment you break down these bonds, a protein is broken down. So to break down these bonds, the enzyme is called peptidase that break down the peptide bond, and hence peptidase is a type of proteolytic enzyme. Does that make sense, Rabia and Iram? Rabia and Aisha, sorry.
Yes.
Yes.
So is it simple now? Understanding chapter 11, the chapter digestion from class 11th?
Yes, sir.
You never ask these questions to your teachers in class 11th? Why are so many words here and there? Why so many types of enzymes? What do they do? I'm sure they must have talked about it. If not, then, you know, I believe that it's a crime to be a boring or a bad teacher. You better don't teach then. Okay. So now why I brought all of this is because amino acids. So now you understand what is amino acids, correct? And where they fit, where they come in the central dogma of biology, right? Now my
Yes,
next
sir.
question to you is, how many types of amino acids are there in human body? Again, make a guess. 400, 500, 29, 87. I want you to say something so that this stays in your memory, that we gave an answer in the class which was so correct that Rahul appreciated, or it was so incorrect that Rahul discussed. So I want you to give answers. How many amino acids are there in human body? If you completely do not know the answer, just make a guess.
400.
400, okay. Aisha?
30 to 40.
30 to 40. Okay. So both are very, very interesting guesses. You know, that just tells that how human psychology works. When we have to make a guess, we always think, okay, no one of you made a guess like 6 lakh. There could be 6 lakh amino acids in a human body. A human body is so big. But if I would have asked you how many cells are in a human body, you would not have said 30 to 40. So you have some sense, you know, inherent logic. That's what makes you human. Always remember that. But that sense is incomplete without knowledge. So there are 22 amino acids discovered so far in human body, out of which only 20 is in your syllabus. The other two are just a little bit modified versions of 2 from the 20. So there are 20 amino acids as far as your business is concerned. Okay, how many amino acids are there?
20.
20. Okay, 20. These 20 amino acids in all multiple combinations make millions and millions of different kind of proteins. We do not know as of now how many total number or different types of protein are present in one single cell, any cell. Every day, you will find that scientists are figuring out one new protein in a new cell. So inside a cell, we don't know. We cannot say with surety that this is the limit. These are the number of types of proteins that are present. Okay. But all the proteins, what we know for sure is that only 20 amino acids make all these proteins. Now, whenever I say something, don't just listen to it and accept it. Ask, how do we know then? How are we sure that there can't be more amino acids, right? Because doing a breakdown study is easier. You take any protein. So what scientists do is whenever they find a new protein, they just take the protein, put some proteolytic enzyme known as peptidase in that protein, and then do something called a spectrophotometry or an analysis, which will tell you about structures of their monomers. And after analyzing thousands and thousands and thousands of proteins, we have figured out that whatever the protein is, it can be broken down into 20 different structures, which are called their monomers, which are amino acids. Phenylalanine is one of the amino acid present in our body. Does that make sense? Story of phenylalanine is the origin. Okay. Shall we move forward?
Yes, sir.
Okay. So phenylalanine inside our body has to be converted into another amino acid called tyrosine, which we do not normally take directly in diet. So tyrosine is a very, very important amino acid in our body. And there are lots of pathways in which tyrosine is required, pathways which form our own enzymes, pathways which form our own muscles, pathways which also make energy from amino acids. So remember, protein is the last fuel that a body burns. So let's say I start starving a normal person. Okay, I'm just giving you some context why this disease is very, very interesting. So in a normal person, there is sugar, carbohydrate, fats, and proteins. Correct? Agree, both of you? In a normal human,
Yes,
there will
sir.
be.
Yes, sir.
You must have studied in your lower classes that carbohydrates are the instant source of energy, right? Have you studied this line? If someone is feeling weak or is very hungry or fainted because of some, lack of energy, what is the first thing you will give the person? A chicken biryani or a Glucon-D? Someone fainted while standing in the sun for a very long time. So what is the first thing you will give that person?
Water.
Glucon-D.
Yeah, water to just make the person conscious, make the person rehydrated. But mostly in that water, if you want to give them with some energy, you put glucose, right? Yes or no? You will not
Yes,
ask the
sir.
person to start eating chicken or curry pakoras, something like that. Okay, or a burger. So let's say glucose is the instant source of energy because carbohydrate is the first thing that the body burns whenever it needs energy. Let's say I'm starving a person. I'm not giving a person food, or a person is on diet or starving himself or herself. Or let's take a situation which is less, torturous. Someone got stuck in a jungle or something, lost the way, and now is starved. So first body will burn all the carbohydrate After that, what it will start burning? Fat or protein?
Fats.
Fats. So it starts burning fat. And finally, at the very end, when body has no other reserves of energy, then it starts breaking down protein and starts making energy from these amino acids. In that case, body starts eating up itself in a way, okay? Because brain wants to stay alive at any cost. If you are losing muscles, but the brain is alive and you are alive, it's a good cost to pay where, because you're not at the very end. So normally in a phenylalanine converts into tones in the body. And for this conversion, anything that happens, for this conversion, there is an enzyme which is important. It is saying that my internet is not stable. Can you hear me, people? Am I audible, people?
Yes, sir.
Okay, perfect. So for this to happen, for phenylalanine to convert into tyrosine, we need an enzyme. Now, if it's an enzyme, its name should end with is, right? And it is converting one type of amino acid, which is phenylalanine, into another type of amino acid, which is tyrosine. So the name of the enzyme, can you guess what should be the name of the enzyme? My idea is that one day you will start guessing these names, even without any prior knowledge, a lot more correctly. That's why I ask you. Can you guess what should be the enzyme's name?
Uh,
Yeah, come on. At
so
the
you're
worst, you
talking
will.
about tyrosine, like the enzyme of tyrosine.
Yeah, so the enzyme is converting phenylalanine. Right, into tyrosine. But yes, come on. So the name of the enzyme is phenylalanine hydroxylase. It again ends with ase. So it adds a hydroxy group on phenylalanine and converts it into a different structure, which we call as tyrosine. So the enzyme also tells about its own function. For your NEET preparations, it might be handy to know that kinase, if there is an enzyme whose name ends with kinase, phosphofructokinase, glucokinase, hexokinase. Okay, kinase means the enzyme adds phosphate group. So if any enzyme adds a phosphate, okay, or removes a phosphate, it's called a kinase enzyme. If an enzyme adds a hydroxy group, it's called a hydroxylase enzyme. So it is reducing the previous structure. Okay, so tyrosine is a more reduced form of alanine. Do you understand? Sorry, phenylalanine. Is it making sense, Aisha, Rabia?
How would you repeat?
Should I repeat from where? Like what part?
Kinase.
Kinase. So kinases are enzymes that adds phosphate group or removes phosphate group. So they deals with phosphate groups onto something. So you will study and you already have studied. Have you studied glycolysis pathway in class 11th?
Yes, sir.
So did you study the enzymes that are responsible when glucose enters in the cell? What happens to that glucose? When it enters into glycolysis, what is the first thing that happens to glucose?
Triose phosphate.
Sorry, come again.
Triose phosphate.
Yeah, triose phosphate comes very late. Before that, glucose becomes glucose 6-phosphate.
Yeah.
Yeah, the first step is glucose comes in and a phosphate group is added on the glucose and it becomes glucose 6-phosphate. The second step is where another phosphate group is added, it becomes glucose 1,6-bisphosphate. And then the third step is where you just convert glucose into fructose and it becomes fructose 1,6-bisphosphate. So the first step where you are adding phosphate on glucose, do you remember what was the name of the enzyme? The name of the enzyme was glucokinase or hexokinase. Hexo because hexa means? I don't remember these things because I have mugged up. I remember these things because they are just simple logic. What does the word hexa means? Hexagon in mathematics.
6.
6. How many carbons do glucose have?
6.
6. C6H12O6 is the formula for glucose, remember?
Yes, sir.
Yes. So hexokinase is a kinase enzyme that adds phosphate on a hexose sugar. And glucose is a hexose sugar. It has 6 carbon. There are also pentose sugar. Can you tell me any sugar which is a pentose sugar, which has only 5 carbons? Ribose is a pentose sugar, which is present in DNA and RNA. Okay. Deoxyribonucleic acid, and RNA is ribonucleic acid. So the ribose is a 5-carbon pentose sugar. And glucose is a 6-carbon hexose sugar. Is it clear? Are these things? So you have to start making sense and linking the dots. Okay. Aisha and Rabia?
Yes, sir.
Perfect. So this phenylalanine hydroxylase converts phenylalanine into tyrosine. And in phenylketonuria, this enzyme is mutated. So this becomes mutated. And after a mutation on this enzyme can no longer function. So there is a single gene which gives rise to this enzyme. And if that gene is mutated, so you write down in phenylketonuria, the gene that codes for the enzyme phenylalanine hydroxylase gets mutated. So there is a mutation on that gene. So only one gene changed. And because of that one gene changing, you understand that phenylalanine will no longer be converted to tyrosine in the body. Yes or no?
Yes, sir.
So what will happen if you are not able to convert phenylalanine into tyrosine in the body, but you are eating things from where you are getting this amino acid? Whenever you are eating a sugar, whenever you are eating a protein, you are getting this enzyme, this amino acid, large quantities, but you are not able to convert it into tyrosine. So what will happen? It will start accumulating in the body. If you can't break something down, it will start accumulating. You will use a lot of it to make your own proteins, but still a lot of it will be just accumulating in the body. And this accumulated phenylalanine, by other ways, starts converting into phenylpyruvic acid. So if this enzyme is not working, then phenylalanine, instead of becoming tyrosine, will become phenyl pyruvic acid. And anything in the acidic form in the body, in the cells, is not good, right? And this phenyl pyruvic acid can start showing a lot of problems. It gets into the brain and causes mental retardation. So just by one single gene getting mutated, the symptoms that you see are multiple phenotypes. One is mental retardation. So kids with phenylketonuria cannot learn, they are mentally retarded, which means their brain is affected. The other is effect of what do you say, skin coloration. Okay, so skin depigmentation. So skin pigmentation is affected, which means it is affecting brain, it is affecting the skin epithelium. These two are very different tissues, right? It also affects the hair follicle. So there is a hair loss in small kids. Hair loss means what is affected? Your hair follicles. Now think about it. Just one single mutation in one single gene, and it controls a lot of different phenotypes: brain, skin epithelium, and hair follicles. So this disorder is an example of pleiotropic inheritance, where one gene controls multiple characters and phenotypes. So in a normal person, this one single gene is helping us with a normal skin color, pigmentation. It's also helping us with healthy hair follicles. It's also helping us with our ability to learn, understand, speak, which is called mental cognitive capability. Does that make sense, both of you?
Yes, sir.
Okay, so what I want from both of you is some commitment, some level of commitment as we go forward. As we are going forward, the fundamentals that you have learned in class 9th, 10th, and 11th are becoming more and more important for our discussions. And if you both are seriously looking forward to gain things in terms of your marks, your career in science, you want to become a doctor, you have to buck up now. Okay, buckle up and start brushing up these concepts. So what I want from you is try to find some time on a daily basis to just open randomly things in your 11th class NCERT biology and just go over it. Okay, just brush it up so that it should not feel like an alien thing to you when we are discussing it in our class 12 syllabus. Are you both willing to give me that commitment, Rabia and Aisha?
Yes, sir.
I'm not
Yes, sir.
asking you to go and start learning chapter wise chapter. No, that's not the motive. Motive is to just keep looking. So what we learned in this chapter in class 11th and keep revising. So what I want is, because at the end of the year, you will be appearing for an entrance exam, right? Mostly people take one attempt at the end of your class 12th. But keeping boards also in mind, we first have to prepare for your subjective and then for the objective exam. And there is no harm knowing more. There's always harm knowing less. Okay. But without you to know more. So just get into of connecting what you learned in class 10th with what you learned in class 11th, and then both of you.
Yes, sir.
Okay, perfect. So I'll stop here. because in the next class I want to pedigree analysis and chromosomal and genetic disorders for this chapter. But I want you today and tomorrow, which is Monday, Tuesday, before you come back on Wednesday, to revise this chapter from beginning, chapter 5, from beginning till here, till finish.